Hi, my name is Jasper Fabius. I'm a PhD student at Utrecht University, and in this lecture I will give an introduction to magnetoencephalography, or MEG, and its use in cognitive neuroscience. So first I'll introduce the general idea behind MEG, then we'll talk about what the signal is that's being recorded with an MEG and how it's actually being recorded. And then I'll show one particular example of how MEG data can be analyzed, which is using multivariate pattern analysis. Okay, so first let's look at the MEG machine, this one. As a subject, you're placed inside it sitting straight up, which is quite comfortable, quite relaxed. It's just a normal chair. And it's at least less annoying than being, for example, in an fMRI experiment where you're placed inside this very narrow tube in a very noisy environment. And for experimenters, it's nice that for subjects, the experience is quite relaxed and they're feeling okay during the experiment. But more importantly, deciding which technique you want to use in your experiment to address a specific empirical question is what are the properties of a specific neuroimaging technique. So for this, I'll show the spatial resolution of a couple of techniques against the temporal resolution. So the temporal resolution, the higher it gets, meaning that you get less information per time unit. And the spatial resolution, the higher it gets, means that you get less information per area. So obviously we would like a technique that is in the lower left corner here with a very high spatial resolution, so very localized information and very fast sampling rates. And there are a couple of techniques that meet these requirements, but all of them require the skulls of subjects to be opened up, which kind of limits the number of volunteers that you could get. So there are other techniques, of course, like fMRI and EEG. MEG is also here. And as you can see, MEG scores quite well in the temporal domain, similar to EEG, but not as good in the spatial domain as compared to, for example, fMRI. And it's said that MEG has a slightly higher spatial resolution than EEG, and we'll talk about it in the next couple of slides why that might be. So in general, MEG is quite a fast technique. So for questions that involve timing, MEG is a very nice neuroimaging technique. So what signal does an MEG measure? For this, we'll have to look at some basic neurophysiology. And we'll consider the neuronal communication between two neurons as an example. There is the presynaptic cell and the postsynaptic cell, and the information is flowing from the presynaptic to the postsynaptic cell. Now, communication starts with an action potential in the presynaptic cell, which leads to the release of neurotransmitters into the synaptic cleft. And these neurotransmitters then bind to receptors in the postsynaptic membrane of the postsynaptic cell, which causes ion channels in this membrane to open or close, leading to a change in the membrane potential of the postsynaptic cell. Because this membrane potential changes, there is also a change in the electrical current in this postsynaptic cell. And this small electrical current also generates a small magnetic field. So if this would be our this arrow would represent our postsynaptic cell, the current would flow this direction, which means that it generates a small magnetic field that flows outward from the electrical current. And this small magnetic field that is generated by our single postsynaptic cell is way too small to be detected by modern MEG systems. But the combined magnetic fields of several neurons that are oriented in the same direction, that is what is being picked up by an MEG. So it's the electrical current that generates a magnetic field, and then combined with of several neurons, this magnetic field is strong enough to be detectable by a modern MEG system. And the benefit of detecting this magnetic field rather than the electrical current directly, which is what EEG does, is that the magnetic field is not distorted by the different conductivity levels by from the brain tissue and the CSF, so the cerebrospinal fluid, and the skull. So that changes the electrical current, but not the magnetic field. Because the magnetic fields flow radially outward from the currents in the postsynaptic neuron, The MEG is most sensitive to the magnetic fields from neurons that are oriented tangentially with respect to the skull. This one. So here's the skull, here's the cerebrospinal fluid, this is the cortex, and this is an example neuron that is oriented tangentially with respect to the skull. So the field is flowing outward like this. And the MEG is less sensitive to neurons that are oriented radially with respect to the skull. And as a side note, the EEG is exactly oppositely sensitive to these two neurons, orientation of neurons. And another addition to this is that MEG is also primarily sensitive to the superficial signals, meaning signals located in the cortical surface, and less sensitive to signals that are generated in deeper brain areas. So this figure illustrates the sensitivity of the MEG to different locations in the brain. The more red, the more sensitive, and the more blue, the less sensitive. Right? So you can see clearly the cortex here is red, and the deeper you go becomes blue, and means that the MEG is much less sensitive to sources that are all the way deep down here in the brain. Okay, so now that we know what the signal is that the MEG uses, we can ask the question how it actually measures these signals. So we already saw this is the machine, but now we're going to look at what is inside the machine. So we're going to look at the sensors. And remember that the magnetic fields that are created by neuronal activity are extremely small. The amplitude of a stimulus-evoked response, which is quite a strong response relatively, is in the order of 100 femtotesla, which is 10 to the power of minus 15. In comparison to the Earth's magnetic field, this is about 100 million times smaller. So this means that we need some good protection from other sources that create magnetic signals, and also we want to be as close to the source of our signal, so as close to the neurons in the subject's brain as possible. So the sensors of an MEG are located in the helmet right here. And it would look something like this if a subject is perfectly oriented in the MEG system. You have a bunch of sensors that are incorporated in the MEG system and are as close to the subject's head as possible. So that's one requirement. We are now as close as we can without opening the skull of our participants. And to get rid of external sources of MEG or of magnetic signals, magnetic fields, the entire system is placed inside a shielded room. So we can eliminate as much noise from outside sources as possible. The part of the MEG that houses all the sensors is the part on top, right here. It's called the DWAR. And schematically, our entire system looks like this. So the square outline is the shielded room, and this part here is the DWAR. And inside the DWAR we find the actual sensors, which are the SQUIDs. And SQUID is an abbreviation for Superconducting Quantum Interference Device. And the SQUIDs then are connected to flux transformers. So the transformers, those are the ones that pick up the magnetic fields. And there are several types of flux transformers. We'll talk about them in the next couple of slides. So there are magnetometers, planar gradiometers, and axial gradiometers. And because the SQUID is a superconducting device, to keep it superconducting, it needs to be in an extremely cold environment. So that's why the entire DWAR is filled with liquid helium. And the liquid helium evaporates slowly, but about every week the DWAR needs to be refilled with another batch of liquid helium. As said, there are three common types of flux transformers. The first one is the magnetometer. It's the most simple design. It consists of a single coil like this, and whatever the single coil picks up is being transmitted to the SQUIDs. Now, because it consists of a single coil, it also makes it more susceptible to noise from outside sources or sources that are further away from the sensor. And to overcome this issue, we have planar gradiometers. These are a little bit more complex. They consist of two coils, or two halves in this case, but you can consider them as two coils, and they are configured like a figure 8. So you have to follow the arrows here. You see that they cross over here, and then this one goes in the other direction. Because of this configuration, this figure 8 shape, the signal that is being transmitted to the squids is actually the difference that these two coils pick up. And the difference between these two coils is large for sources that are close to the center and very small for sources that are further away, because the sources that are further away from the sensor will generate basically the same signal in both coils. The same goes for the axial gradiometers. Rather than being two coils that are laid down next to each other, here we have two coils that are above each other, as you can see here. So the signal that is being transmitted by these gradiometers, by the axial gradiometers, is the difference between these two. So a source that is coming from the brain, like let's say down here, will be stronger in this coil than in this coil. And if you want to know more about this, there's a very nice lecture by Stephen Whitmarsh on YouTube. I put the link down here, and he explains very clearly how these different types of flux transformers are sensitive to different sources or different distance sources. And yeah, I recommend this lecture if you want to know more about this. Because of the different properties of these types of flux transformers, different manufacturers have put different configurations of these flux transformers into their systems. For example, the 4D neuroimaging only has magnetometers. The Yokogawa and the CTF 275 both only have axial gradiometers, and the electroneural mag has both magnetometers and planar gradiometers. Okay, so to give an overview of what we've covered so far, the MEG has a good temporal resolution, sampling more than every 1 millisecond, or at least it can go up to that resolution, sampling rates of over 1000 Hertz. And the signal that's being picked up is related to the postsynaptic current, and then the magnetic fields that are created by that is what we're actually using with an MEG. And these magnetic fields are then picked up using the squids and the flux transformers. And there are different types of flux transformers that all have these different kind of configurations and making them sensitive to different distance sources or different distances. So because MEG is a very fast neuroimaging technique, I will give one example of a way of analyzing MEG data that really exploits this fast sampling rate and this fast property of the MEG. The example I'll use is time-resolved multivariate pattern analysis. It's a quite recently developed technique, and it can be used to answer questions like, how does the brain process visual stimuli? And because we think that there are multiple steps in processing a visual stimulus, for example, we start with different contrasts on the retina, and in cortical areas then we can detect edges, and then there are several other steps all the way up to a point where we can, as humans, name specific objects, like a chair or a couch, all these things. We can name them, and they all start from a very basic image on the retina. So with time-resolved multi-pattern analysis, we can try to figure out where and when, after the onset of a stimulus, information in the brain is actually giving us something that can tell us whether it's a couch or a chair. And the example study that I'll use here is going to be focusing on animate versus inanimate objects. So when can you tell, based on the signals in the brain, that a specific image that is being shown to a subject contained an animate object or an inanimate object? And this entire explanation can be found in a tutorial paper on time-resolved MVPA, as it's abbreviated, by Tel Grootslagers and his colleagues that was published last year in Journal of Cognitive Neuroscience. So in this paper they also use an example study where the presentation of images was like this. So they had a specific object presented very briefly, so it was flashed 66 milliseconds, then followed by a blank screen in which the subject had to respond, was the image that you just saw an animate or an inanimate object, and then the next image was shown. And these images, they were drawn from a collection where you had animate objects and inanimate objects. So these are all different species of animals, and these are all different inanimate objects. So the task is just tell me whether the picture that you saw was animate or inanimate. Now, while the participant is doing this, they are inside the MEG. So the MEG is actually recording the brain signals while participants are doing this task. So here they are. This participant is actually looking at a face here, which would be an animate object. And all the sensors in the MEG are working, which gives us quite a lot of data. So in the output we could see something like this, and for every sensor here there is one line over time recording the magnetic fields. So you can see this time is 1 second, and you can see all these squiggly lines. So how to make sense of all these squiggly lines in relation to the stimuli that have been presented to the participant? Of course, what we could do is take a look at the average activity for each sensor, and then we separate the average activity for all the animate objects versus all the inanimate objects. So schematically, this is what we then did. We presented our images to our subjects and measured with the EEG the responses that were evoked by these different images in their brains. And we measured that with the MEG. Now here, all the blue circles could be our animate objects, and the red squares are then the inanimate objects. So let's consider one particular sensor, and let's look at the average activity evoked by these different categories. So the average activity for class B, or our inanimate object, and class A, our animate object, over time looks like this. So let's assume the stimulus was being presented right about here. So first there is not much response, and then after a short while we can see that both categories evoke this strong peak in the magnetic fields that are being recorded by our MEG system, and then down a little bit, and then back to baseline. Now if we would look at this, we could say, okay, let's pick a particular time point right here, and now let's look at the distribution. So we have multiple images, so the average activity is the red line for inanimate objects, but there is some variation around it. And if we would plot the variation, so we cut through the signals here, through the averages, and plot them on a different axis, we have all our inanimate objects, the red line, and all our animate objects, the blue line. So this particular sensor responds slightly stronger to our animate objects than inanimate objects. But there is quite some variation, and the two are not that distinct, right? The two distributions are not separated by a big distance. They overlap quite a bit. So we could also look at a different channel. This one, for example, has a slightly different pattern. If we look here, we can see that the red is slightly higher than the blue, which means that it responds slightly stronger to inanimate than animate objects. Now I'm going to plot them again, all the variation taken into account. There is this difference between the two, and now the strength of the signal goes upwards, and we can see that it responds slightly stronger to inanimate objects than animate objects. But again, the separation between the two is not that big. Now what MVPA tries to do, so multivariate pattern analysis, is to combine these two signals and see whether if we combine them, we can have a stronger separation between animate blue and inanimate in red objects. So if we do that, we can see here, as an example at least, that we might be able to find a much stronger separation if we combine these two signals. And if we combine these two, then we can get a separation like this one. And you can see that the separation between these two distributions is much larger than if we look at the univariate distributions. So the sensitivity of combining multiple sensors at the same time can increase the detectability of differences between the two categories. Here we only combined 2 channels, and it already improved our discriminability between the 2 categories that we had in our experiment. Of course, we can also combine all the other sensors that we have, and as you remember, usually we have more than 100 sensors to combine. So if we would do that, we can look at the combined activity pattern of all these sensors at the same time, and we can consider this specific pattern as the physical manifestation of a mental state induced by the experimental condition. In practice, we would use all the sensors of the MEG at the same time, so we would look at the pattern across all the sensors at a particular time point. For simplicity here, I'll only use 2 sensors because it's easier to visualize and easier to explain with 2. So we have sensor 1 and we have sensor 2, and each image that we showed, was it an animate object or an inanimate object? The inanimate were red squares, animate blue circles, evoked a particular response in sensor 1 and a particular response in sensor 2. And this pattern of activity was at one particular time point. And remember, we can sample at a very high rate. So this is, let's say, the activity pattern at time point 100 milliseconds after stimulus onset. Now with MVPA, we take apart a subset of trials, we put them there. For all the other trials, we use them to train a classifier, and a classifier is going to make a decision boundary that best separates our 2 classes based on the activity that they evoked in sensor 2 and sensor 1. So if we would separate the blue circles from the red squares, the best separation is this diagonal line, right? So most of the inanimate objects are below and all of the animate objects are above, and there's one inanimate object above, but this is the optimal decision boundary. Now we trained our classifier on this subset of trials, and we're going to evaluate its performance using the remainder of trials that we took apart earlier. So if we do that and we apply our decision boundary to these trials based on the activity that they evoked in sensor 1 and sensor 2, we can see that our classifier makes 1 error, but 3 of them were classified correctly because below the line should be inanimate, so all the red squares, and above the line should be animate. Now our classifier classified this little point as an inanimate object even though it was an animate object. So that means that the classifier performance at this time point, based on sensor 1 and sensor 2, is 75% correct, which is not bad. Most of them were classified correctly. Because this is one time point, we can also repeat this for all the other time points in our data. And then we can look at how the classification performance evolves. If there is no distinct pattern between animate and inanimate, we would expect that classification performance is 50%. It's a chance level. If, however, there is information in the sensor 1 and sensor 2 data that is specific for animate versus inanimate objects, we would expect that classification performance is above chance, like it is here. And now we can see how classification performance evolves by doing this entire procedure for every time point. And then we can make a graph like this. And let's zoom in on this. On the y-axis we have classification accuracy, which is the percentage of trials that were classified correctly from the test set based on the classifier trained on the other subset of trials. And on the x-axis here we have time since stimulus onset, and the time when the stimulus was actually on the screen is depicted here in gray, the bar on the axis. So with time-resolved multivariate pattern analysis, we can look at the temporal dynamics of brain representations as picked up by the MEG. As we can see here, before stimulus onset, there is no clear distinction between these animate and inanimate objects. But very rapidly after stimulus onset, there seems to be some difference, although not very clear, because classifier accuracy does not exceed 55% accurate. Then dips down a bit, and for the remainder of the epoch, it's definitely above chance. So there's definitely a distinction between animate and inanimate objects. And that's really the strength of time-resolved MVPA, which you can do with MEG. You can actually also do this with EEG, but for now it's more common to do it with MEG for some reason. And being able to study the brain dynamics over time like this is really an advantage of MEG over, for example, fMRI. This concludes my introduction to magnetoencephalography. We've covered several topics of MEG. First, the signal that we use, which is the magnetic field that is created by the postsynaptic membrane potential, and specifically those of similarly oriented groups of neurons. These magnetic fields are picked up by sensors. We have different types of flux transformers. We've discussed those. And the primary advantage of MEG is to study the high temporal dynamics of brain processes with an analysis such as the multivariate pattern analysis. Okay, thank you for your attention.